Hello, this is Miss Dagenford, and we're going to be looking at the receptors of the B and the T cells. And the first thing that we're going to look at would be the antigen binding site. And you can see that right here on the end. And the antigen binding means the specificity for all of the B and the T cells. It is made of the variable regions that are found from the light chain and then the heavy chain. You can see them right here that I'm circling. And you notice that the two ends of the light chain and the heavy chain create that shape. So if we were to model this to the left here, we might see a B cell for influenza B, and the receptor might look like this. And then over here, we might see the measles B cell, and the receptor for it might look like that. And that would create the receptor. They are similar in shape to the antibodies that are released by B cells, but you'll notice that they are anchored. So down here on the transmembrane region, the receptor is anchored to the membrane, whereas the antibodies are released. The one thing that they do have in common is the antigen binding site is going to be matching that epitope that is on the pathogen, and that's what gives the specificity. For each specific epitope that might be on the pathogen, you might make different antibodies. So you'll notice the antibody A and antibody B and antibody C are all created by the different variable regions. So that's the receptor for B cells. If we swing down to the T cells, you'll notice some similarities. We have the antigen binding site, again creating that specificity, again created by the variable regions. You'll notice the difference is that we don't have the Y shape. We just have these two equal size polypeptide chains. And again, they are anchored in the transmembrane by the transmembrane region. What we will notice with the T cells is the interaction between the T cell and the antigen presenting cell. And we will get into this in more detail, but it's this antigen that is recognized by the T cell. And you'll notice that this antigen gets onto this pocket of this MHC molecule. That antigen is going to be complementary to the receptors. Similar to the active site, similar to the receptors that we've noticed with regulation, those proteins have to be the right shape. If we go to the MHC molecules are going to be found on all cells. That would be the MHC1 protein. Now, the MHC stands for major histocompatibility complex. And this word can be quite frustrating at first, but major just simply means it's a significant part. Histo refers to tissues, and the compatibility refers to the understanding that this is what makes us us. This is what makes us individual. So for instance, person A might have an MHC molecule that might be shaped like that. And then person B, the MHC molecule, might be ever slightly different in shape. This is what leads to tissue rejection in organ transplants, where the immune system recognizes the fact that all these tissues are different. So person's A tissues would display this MHC molecule, and if you were to transplant an organ by person B, that immune system recognizes it as foreign. So the MHC1 protein is displayed by all cells that have a nucleus. This would then exclude red blood cells, but they have their own ABO blood group. The purpose of the MHC1 is to display antigens. It's like the ID badge holder that you might carry for your school badge. Now, they display what are called endogenous antigen. And endogenous, just think of the fact that endo means from the inside. So you're going to see two things happening here. They could display normal cell proteins. And if this is the case, then what we're going to see would be the fact that the proteins, let's model this over to the right here, you'd have your nucleus and the rough ER that's surrounding the cell. And the rough ER is going to bring in a protein that's going to be part of that cell's cytoplasm, just a normal cell protein, and it's going to be brought in. And what we're going to notice is that a vesicle pinches off from that rough ER, and this is where we're going to find our MHC protein. And the MHC protein is going to be embedded in that vesicle membrane. The cell will attach that endogenous antigen onto it. The vesicle will travel to the cell membrane where it fuses, and MHC molecule will become part of that cell membrane, and it's displaying that antigen, much like that ID badge. Now, if this is the case and it's a normal cell protein, then basically this is establishing the normalcy. This is a cell status. It's saying, I'm okay. This is your typical throat cell or your pancreas cell or your intestinal cell. It's just declaring its identity. Now, the second thing that might happen is this might be a viral antigen. And this is occurring because the virus could be reproducing inside of the cell. So if we sneak over to the right here, let's model that, in which case here might be a throat cell. And again, we have our nucleus down at the bottom and the rough ER. In the case of a cell that has been infected by a virus, you're going to have these viral particles being made and potentially being shed. But while it's being made, you have these viral proteins being just within the cytoplasm of the cell. And what the cell can do is, again, take that little vesicle in the rough ER with the MHC1 protein, and then it's going to take the viral antigen and display it on the surface of the cell in a similar process. So you have MHC1 again on the surface. This time you're going to be displaying the viral antigen. In this way, the cell now declares the fact that I'm infected. And this now declares something that is hiding in the inside of the cell. It's now declaring that to the outside of the cell. And then the immune system can respond to that. Now, the MHC2 is slightly, well, it is similar in that it displays antigens. This is going to be found on what we call antigen-presenting cells. Now, thankfully, the name says it all. These are cells that present antigens. We have two that are generalists. They basically engulf anything that they don't recognize, macrophages and dendritic cells. And these wander around the body, and if they don't recognize you, they eat you, they consume you. We also have B cells that could be antigen-presenting cells, but their job at that point is just to help stimulate and activate other immune cells. So the way these guys work, they display exogenous antigens. And the exogenous antigens, they come from the outside, and you can see the name exo. So they come from the outside. So the way this is going to work is you've got these generalists, and they kind of wander around, and if they see a pathogen, they operate very simply, engulf it. And so we've done this before. Here's our little pathogen, and it's going to be brought inside the cell. And then it's going to be digested by a lysosome. And as it's digested by the lysosome, there's our little lysosome, and into all of its parts. And some of those parts would include those viral or pathogenic antigens. Now, in a similar way, we can take that exogenous antigen and also plug it into our MHC molecule. This time it's the MHC2, and it's going to go to our cell membrane. This time the job here is to display display the enemy of the body. So if this is the pathogen, this antigen presenting cell is then going to go off to find the appropriate B cell, the appropriate T cell, and of those T cells, we're going to get our helper T cell, and then the cytotoxic T cell. We're going to activate them. And their job is to essentially say, this is the enemy. Now remember, you've got thousands upon thousands of different B cells and T cells, most of whom are dormant, naive, and so you really only want to call up the specific B and the T cell. So the MHC2, that's the purpose. It's going to be able to seek out the right B and T cell by matching to that receptor, and then that's going to set off a signaling process. So I hope that provides a little bit of background about how the cells are unique, how the cells respond, and how we create the specificity. Thank you so much.